I'm here doing a lab for my AP Biology class, and our discussion in class has been on cellular respiration. So therefore we had to make up our own lab, which was pretty interesting actually, about cellular respiration and insufficiency. So therefore we chose to do temperature. So we have ranging temperatures. Over here we have hot, warm, room temperature, and cold. We're going to see how cellular respiration is in those different temperatures. Our hypothesis is if the temperature is increased, the amount of carbon dioxide in the fermentation tube will be greater because cellular respiration is more efficient in a hotter climate. So our prediction is that this one will produce the most carbon dioxide. And what that means is that the enzymes in the fermentation tube, which is yeast and sugar mixture, will produce more carbon dioxide because they are reacting more efficiently. So therefore, in the fermentation tube, the buildup of carbon dioxide will be greater. And the opposite pole in the refrigerator over there is going to be less. So enzymes will react slower, not as efficiently, and therefore the carbon dioxide buildup will be a lot less. We used yeast and 4 fermentation tubes, a ruler, sugar, water, measuring cups, 5 beakers, a heat source, and a cold source. So the basic procedure of this lab is simple. First, more importantly, you have to set everything up properly, mix the solution, have the freezer ready to go, which I close that. So yeah, the setup's important. Make sure everything is ready to go, the right temperatures are set. You pour the stuff into the fermentation tubes, but after that, simply you just have to watch it and use your ruler here, another important piece of equipment for this lab, and just measure the carbon dioxide buildup. Alright, so right now I'm pouring the mixture that we mixed earlier, the yeast and sugar solution, into the fermentation tube. Pour it in like that. Pretty simple. You want it up here. Let it up. There you go. So we're going to be doing that 4 times because we have 4 different temperatures. So, same thing. Go around and place your fermentation tubes into the waters. Just make sure it's not flooding into the fermentation tubes. Alright. Next one, which is about at yeah, that's hot. That's cold. Put in the fridge now. So every 5 minutes, and it's been 5 minutes now, you're going to measure the carbon dioxide buildup. So this is kind of tricky because it's right there. It's kind of covered in water. So basically, the enzymes work very quickly, and that fermentation tube is all the way down. So cellular respiration occurred very quickly in the hot one as we predicted. In the warm water, not quite as much, but still quite the carbon dioxide buildup there. Room temperature, a little bit less, and I'm going to guess in the freezer, in the refrigerator, it's going to be the least, so let's check it out. Yeah, but it's barely moved down the tube at all. So keep on doing that at the 10th minute, 15, all the way up to 30. Make sure the temperature is right, so this one right here, you can bring the camera closer, it's right around 98, it kind of fluctuates up and down, but right around 98 degrees Celsius. Over here, so we're 95, this one's the 8. This one is right around 48, it's kind of down right now, but I just turned it up a little bit, but it's not a big deal as long as they're ranging within the close proximity of that number, because the temperatures are varying greatly, so a little hindrance in the number is fine. Alright, so now that the lab is done, careful with this one, it's hot. And that's raising up, that's fine though. Just be careful with the hot plate. Alright, so when you're all done, just go to the beakers, take out the fermentation tubes, so you can just set them over there. Make sure your hot plates are turned down. Just clean up the thing so you can just take it. Here. Might be a little bit hot, so just be careful. You just pour it out, etc. Just keep on going around and doing that with other ones. Oh, it's not put back on there, so a little bit warm. But yeah, just go around, just clean up your lab, and then you can sit down with your teammates for the lab or just by yourself and analyze the results. Once you've gotten all your results down, all your data collected, you can analyze that with your team by yourself. I guess if we had time at the end of class, we would have done it then. Otherwise, just do it at home. It's pretty simple. Just kind of look at your results. At the 3 degrees Celsius, it was 0 the whole time. At 20 degrees Celsius, it was a gradual increase, ranging from anywhere from 4 to 7 per 5 minutes of carbon dioxide buildup, which is measured in millimeters. So we just kind of millimeters. And warm hot bath was rapid at the beginning with a big 44 millimeter jump, and then it was somewhat gradual for a while, and then it kind of started to slow down. This one was different though, and we'll get to that. Alright, so this is the outlier, and our hypothesis was correct, but with a little bit of a variation in it. Because of that, at first it was very rapid and the most efficient. The carbon dioxide built up extremely rapidly, and yeah, it was the most efficient at the beginning. This one was a close second. However, at about the 20th mark, this one started going down. And that's probably just human error because the thing was tilting, but basically the enzymes were not active anymore. So we're predicting that they denatured or were completely used up, the cellular respiration process, so it just stopped. This one, however, also a warm temperature, that it was at a warm climate, and kept on increasing fast at first and then more gradual. So basically our hypothesis was correct, that in the fermentation tube we'll have a greater buildup of carbon dioxide if it's in a warmer temperature.